pursuit of continuous quality education.
Living up to DepEd's mandate of no student should be left behind.
From the home of Sarongbangi,
The land of Oragons. This is Santo Domingo National High School. School on Air.
School on Air.
Good afternoon to my curious, motivated, and creative Grade 10 young scientists. A special mention to my Grade 10 Einstein, Grade 10 Dalton, and Grade 10 Newton. I am glad to be with you again. This is your radio teacher in science, Ma'am France. In my science class, no mysteries will be left unsolved. Ask, thinker, and explore. You are a brilliant scientist in your own laboratory, now at home. At this point, please get your learning activity sheet or LAS in Science 10, Module 4, entitled DNA Structure and Chromosomal Mutation. It will serve as short guide for our discussion for today. Please have a ball pen and a piece of paper so that you can jot down notes while I am discussing the topic. Near the end of this episode, you will be using your mobile data for sending your answer to the questions and activities. For those who do not have their cell phone with mobile data, don't worry, because you can send your answer in the scheduled distribution and retrieval of module. Make sure you are in a comfortable place, far from noise, so that you can hear me loud and clear. Are you ready to learn? I hope you are eager as how you get up this bed this morning. In grade 9, you were able to describe the structure of the DNA and make models of DNA molecule. You also learned that the genes in their DNA influence their characteristics. What you will learn today is how a protein is made using information from DNA. To be able to function properly, all cells require some form of instructions and codes for making material in the cell. That code is DNA. DNA stands for deoxyribonucleic acid. D stands for the name of sugar, which is deoxyribose, and Na for nucleic acid. DNA contains the information that determines inherited characteristics. It has the code for making protein. Just as DNA, RNA is also an important biomolecule. RNA stands for ribonucleic acid. R stands for the name of sugar, which is ribose, and N and A for nucleic acid. Let us compare DNA and RNA. What are the components of DNA and RNA molecule? Exactly. The DNA and RNA are made up of three components, namely sugar, phosphate group, and nitrogenous base. What is the structural difference between DNA and RNA? You're right. DNA and RNA are different in the following. 1. DNA is double-stranded, while RNA is single-stranded. 2. The sugar in DNA is deoxyribose, while that in RNA is ribose. And 3rd, the nitrogen bases in DNA are adenine, A in symbol, guanine, G in symbol, thymine, T in symbol, and cytosine, C in symbol. In RNA, the nitrogen bases are adenine, A, guanine, G, cytosine, C, and uracil, U in symbol. What nitrogenous base is found in RNA but not in DNA? Correct. Uracil is found in RNA but not in DNA. A DNA molecule consists of two strands of nucleotides composed of sugar, phosphate, and nitrogenous bases that pair through hydrogen bonds. The paired strands form a twisted zipper shape called a double helix. Meanwhile, the eukaryotic cell has the so-called true nucleus, which means that its nuclear materials are enclosed by a nuclear membrane. The DNA is found in the nucleus of the cell, while the RNA is found in the cytoplasm. RNA molecule is single-stranded and is composed of nucleotides. There are three major types of RNA: mRNA or the messenger RNA, rRNA or the ribosomal RNA, and tRNA or the transfer RNA. To make proteins, the messenger RNA or the mRNA is assembled inside the nucleus through transcription. The coded genetic information carried by our mRNA is translated through protein synthesis using transfer RNA or the tRNA. In DNA, adenine bonds with thymine and cytosine bonds with guanine. In RNA, adenine bonds with uracil and cytosine bonds with guanine. I repeat, in DNA, adenine bonds with thymine and cytosine bonds with guanine. In RNA, adenine bonds with uracil and cytosine bonds with guanine. So, what is the central dogma of molecular biology? Or should I say, the central dogma of life? Because it explains how you and I take the genetic information from our parents and pass it on to the next generations. DNA makes RNA makes protein is the central dogma of genetic information. I repeat, DNA makes RNA makes protein is the central dogma of genetic information. Let us explain this concept a little further. DNA and RNA are nucleic acid, which are made up of nucleotides, and proteins are made up of amino acids. DNA copies itself in a process called replication. The DNA is copied into RNA in a process called transcription, and then finally uses the information in RNA to synthesize protein in a process called translation. This whole process is the central dogma. Let us first analyze how DNA replicates, or how DNA makes DNA. A DNA unzips to form two strands. As the molecule unzips, the base pairs separate. Each single strand of DNA then picks up bases present in the cell's cytoplasm. In this way, two complete molecules of DNA are created. Each new DNA molecule has the same order of base pair as the original. The copying process is called replication. Why does DNA replicate? Good! To produce a copy of the genetic material that will be transferred to new cells during mitosis or to new gametes during meiosis. Which nitrogen bases fit together to make base pairs? Adenine and thymine fit together, and guanine and cytosine fit together. I repeat, adenine and thymine fit together, and guanine and cytosine fit together. When a DNA molecule unzips to form two strands, what is added to each strand? What is produced? Complementary bases. Complementary bases attach to the exposed bases on the strands so that two complete molecules of DNA are produced. How is the gene in the DNA coded? It is usually coded by a particular sequence of base triplet called codons. In what form does a gene carry information? A gene carries information through the sequence of nitrogenous bases that may code for RNA or amino acids in proteins. What do you think is the role of RNA in making proteins in the cell? Can you imagine a car being assembled in a car factory? By way of analogy, different cars are being built in many simple steps. Engineers tell workers how to make cars, and the workers follow directions to build the cars. Suppliers bring parts to the factory so they can be installed in the car. Protein production is similar to car production. It is the role of the DNA to provide workers with instruction for making the proteins, and the workers build the proteins. Other workers, known as amino acids, bring part to the factory. RNA molecules, or the workers for the protein synthesis, get the instructions for the DNA on how the protein should be assembled. Do you know how the information in DNA, which is found in the nucleus, move to the ribosome in the cytoplasm? The events in the process of transcription can be read in your LAS on page 1. The following events can help you understand the process of transcription. Step 1. Ribonucleic acid polymerase enzyme binds and opens the DNA molecule that will be transcribed. Step 2. As the DNA molecule opens, the RNA polymerase slides along the DNA strand and links free RNA nucleotides that pair with the nitrogenous bases of the complementary DNA strand. Hence, if the sequence of bases on the DNA strand were CCG, TTA, CAT, the sequence of bases on the RNA strand would be GGC, AAU, GUA. Step 3. When the process of base pairing is completed, the RNA molecule breaks away as the DNA strands rejoin. The RNA leaves the molecule, or the nucleus rather, and goes to the cytoplasm. Let us go back to the given example of the DNA strand. CCG, TTA, CAT. Please follow. Let us go back to the given example of the DNA strand. CCG, TTA, CAT. How does the sequence on the RNA strand became GGC, AAU, GUA? Going back on the concept of base pairing in DNA, adenine bonds with thymine and cytosine bonds with guanine. In RNA, adenine bonds with uracil and cytosine bonds with guanine. So, the DNA codon CCG, cytosine, guanine, when transcribed to RNA codon became GGC, guanine, cytosine, because cytosine bonds with guanine. On the other hand, DNA codon TTA became an RNA codon AAU because T, which is thymine, was transcribed to A or adenine. Since thymine is not a nucleotide of RNA and adenine was transcribed into uracil or U, since both are nucleotide of RNA and binds together. Lastly, CAT became GUA because cytosine bonds with guanine, adenine to uracil, and thymine to adenine. Now, let us try another example. The following is the base sequence on one strand of a DNA molecule AAT, GCC, AGT, GGT. If transcribed into an mRNA, what would be the resulting strand? I will give you 30 seconds to figure out the answer. Again, let us try this example. The following is the base sequence on one strand of a DNA molecule. AAT, GCC, AGT, GGT. If transcribed into an mRNA, what would be the resulting strand? I will give you 30 seconds to figure out the answer. The DNA strand AAT, GCC, AGT, GGT, if transcribed to mRNA, will be UUA, CGG, UCA, CCA. Did you get the correct answer? I hope so. If not, send me a question about the process of transcription in my messenger. The DNA directs the production of protein and determines the formation of mRNA. The order of bases of mRNA determines the protein synthesized. Proteins control the activities of the cell, as well as the life of the organism. But how does DNA make a unique protein that will perform a special function? Would you like to find out how the message of the mRNA is translated into proteins? In translation, each set of 3 nucleotides in an mRNA molecule codes for 1 amino acid in a protein. This explains why each set of 3 nucleotides in the mRNA is called a codon. Each codon specifies a particular amino acid. For example, the first codon, which is cytosine, guanine, uracil, CGU, instructs the ribosome to put the amino acid ARG or arginine in the protein. The sequence of codons in the mRNA determines the sequence of amino acids in the protein. But how are the right amino acids added in the right sequence to match the sequence of codons in the mRNA? The following events in translation can help you understand the process. Step 1. As translation begins, mRNA binds to a ribosome. Then, tRNA molecules, each carrying a specific amino acid, approach the ribosome. The tRNA anticodon pairs with the first mRNA, start codon, arginine, uracil, guanine, or the AUG, that is the start codon, to form the initiation complex. The 2 molecules temporarily join together. Step 2. Usually, the first codon on mRNA is AUG, which codes for the amino acid methionine. AUG signals the start of protein synthesis. Then, the ribosome slides along the mRNA to the next codon. Step 3. A new tRNA molecule carrying an amino acid pairs with the second mRNA codon. Step 4. When the first and second amino acid are in place, an enzyme joins them by forming a peptide bond between them. Step 5. As the process continues, a chain of amino acid is formed until the ribosome reaches a stop codon. Example of stop codons are UAA, UAG, UGA on the mRNA strand. The polypeptide chain is released. Protein synthesis is now complete. Did you now understand how protein is synthesized? Congratulations, you made it! For those who still has a blurry picture of protein production, you can send me a question on my messenger. Now, let us analyze the given example in your LAS on page 2. Please open your LAS on page 2. Direction. Use your codon chart to determine the amino acid sequence. Your codon chart can be found on page 3 of your LAS. Remember to read through the strand and only start on AUG and stop when it tells you to stop. Follow the example given in your LAS. Please refer on your LAS. Given DNA strand is TAC, CTC, CGG, TGG, GTG, CTT, GTC, TGT, ATC, CTT, CTC, AGT, and ATC. Given mRNA is AUG, GAG, GCC, ACC, CAC, GAA, CAG, ACA, UAG, GAA, GAG, UCA, UAG. Synthesized protein. Start, glutamic acid, alanine, threonine, histidine, glutamic acid, glutamine, threonine, and stop. Let us go back to the given example. Given DNA strand is TAC, CTC, and so on. Some of you would ask, how did mRNA become AUG, GAG, GCC, and so on? Again, remember the base pairing. Example, TAC became AUG since T is paired to A to U, and C to G. Also, CTC became GAG because C is paired to G. T to A, C to G, and so on. And how about the synthesized protein? How did it turn out to be start, glutamic acid, alanine, threonine, until the stop? Let us look at the transcribe mRNA, which is AUG, GAG, GCC, and so on, to determine the synthesized protein. You will use the table on page 3. Again, we are going to look at the transcribed mRNA, which is AUG, GAG, GCC, and so on. To determine the synthesized protein, you will use the table on page 3. AUG is M in the table, which represents methionine or initiation or start codon. GAG is represented by E in the table or GLU for glutamic acid. GCC is represented by A or ALA for alanine. ACC represented by T or THRE for threonine. CAC is represented by H or hist for histidine. GAA represented by E or glue for glutamic acid. CAG represented by Q or glue for glutamine. ACA represented by T or threo for threonine. And UAG represented by stop for stop codon. Did you now get it? I hope so. If not, send me again your questions in my messenger. Now, please try this short activity. Get your paper and ball pen and find out the mRNA sequence and synthesized protein from this DNA sequence. A, G, A. C, T, T. A, T, C. I repeat. The DNA sequence is A, G, A, C, T, T, A, T, C. Again, let us try to find the mRNA sequence and synthesize protein in the given DNA sequence. A, G, A, C, T, T, A, T, C. Good job! The mRNA sequence is UCU. GAA, UAG. The synthesized protein is SE, stop, or serine glutamic acid, stop codon. I repeat, the DNA sequence given is A, G, A, C, T, T, A, T, C. You are asked to find for the mRNA sequence, and that is U, A to U, G to C, A to U. So it's U, C, U, G, A, U, A, G. And the synthesized protein is S, E, stop. S for serine, E for glutamic acid, and stop codon. Now, may I leave you problem number 1 on page 3 of your LAS. Please write problem number 2 in your paper. Again, write problem number 2 in your paper. You will find the mRNA and synthesized protein. The DNA sequence will be TAC, ATG, GAC, GAG, GAT, I repeat. TAC, ATG, GAC, GAG, GAT, TCCA, TCC, ACA, ATT. Again, I will leave you problem number 1 on page 3 of your LAS. Please write problem number 2 in your paper. You will find, or you are going to find, the mRNA and synthesized protein. The DNA sequence will be TAC, ATG, GAC, GAG, GAT, CCA, TCC, ACA, ATT. Please submit your answer in the 2 problems in my messenger after an hour. For those who will not be able to submit their answers in my messenger, you can just submit it in the scheduled distribution and retrieval of modules. I hope you learned something today. Thank you for listening. Please send me your questions and clarifications on our lesson for today. I will be more than willing to answer those queries. Thank you. It has been a fruitful day to us. Another mystery has been solved. This has been your science radio teacher saying, in my class, no mysteries will be left unsolved. Ask, thinker, and explore. You are a brilliant scientist in your own laboratory, now at home. Goodbye and happy learning!